Hello students, the next question is on express the process of protoplast culture or describe the techniques of plantlets generations from protoplast. So protoplast introduction, as you write from questions 1, the same information you have to write in these questions. Then isolation of protoplast. This isolation of protoplast will be text or it will be happenings by using the two methods, that is mechanical method and enzymatic method. In enzymatic method, two methods are used. One is the single enzymatic methods and double enzymatic method. That methods I have already discussed in previous videos. So let's talk about the process or techniques of the protoplast culture. So first you have to take the leaf material. This leaf material is sterilized, then peeling off and it cut into these segments. Then plasmolysis process takes place with the help of mechanical or enzymatic methods. Then this material will be filtrated, centrifugation carried out, and at last the inoculations of the protoplast is happening. So fusion of the gene mutation, addition or addition, that is protoplast is the or somatic hybridization, manipulation of protoplast, or it is the fusion of the protoplast, that is known as the somatic hybridizations. Also the manipulation in protoplast are carried out. Also the mutations in protoplast we can carried out by or in this technique. Also we can addition of some organelles in protoplast, that's what also we can do on this protoplast. So all these things we can create on or we can do with the protoplast, and then we can incubations transfer these protoplast on the MS medium. Then it will be kept for incubations. Then we can form, and then on that material callus will be formed. This callus or the for the shoot from the callus to shoot initiation or shoot formation, we can add on this medium with 1 BAP medium. Then for root initiation we can use the cytokinin medium. Then after development of root and shoot, the plantlets will be formed. Then these plantlets will be hardening for the greenhouse, and then slowly this plant will be exposed or released for the general cultivations. So this is all happening in the protoplast culture. First in that first part is the separation of protoplast, then second part is the treatment of this protoplast, and third part is the incubation inoculation and incubations of this culture. So on this way we can got the plantlets. So first step is the isolation of protoplast. That procedure we talk already on previous videos. Then second step is the protoplast culture. So after isolation of this protoplast, it may be somatic hybridization or manipulation or induced mutations or cell organelle additional before culture. So जब हमें प्रोटोप्लास्ट हम सेपरेट करते उसके बाद उसके साथ उससे यदि हम टू प्रोटोप्लास्ट का फ्यूजन भी कर सकते हैं, उसमें कुछ मैनिपुलेशन कर सकते हैं, मतलब उस पर कुछ म्यूटेशन कर सकते हैं, उसमें कुछ चेंजेस कर सकते हैं, कुछ ऑर्गेनेलिज्म उसमें ऐड कर सकते हैं. सो सोमेटिक हाइब्रिडाइजेशन किसे बोलते हैं? तो यदि हम दो प्रोटोप्लास्ट का फ्यूजन करते हैं वहाँ पे, तो उसे सोमेटिक हाइब्रिडाइजेशन बोला जाता है. तो सोमेटिक हाइब्रिडाइजेशन फ्रॉम द प्रोटोप्लास्ट ऑफ द सेम प्लांट और डिफरेंट प्लांट्स का हम वहाँ पे यूज़ कर सकते हैं. दिस फ्यूजन ऑफ प्रोटोप्लास्ट इज carried out by mechanical or chemical method. This method also carried by the fine माइक्रो कैपिलरीज के मदद से वो हम प्रोसीजर कर सकते हैं. उसमें हम जो प्रोटोप्लास्ट है, प्रोटोप्लास्ट का फ्यूजन करेंगे with the help of some chemical methods that will be carried by the polyethylene glycol or mixture of the fusiogens. तो उससे हम प्रोटोप्लास्ट का फ्यूजन कर सकते हैं और फिर उसका हम इनोकुलेशन न्यूट्रिएंट मीडियम पे कर सकते हैं. देन सेकंड है जीन मैनिपुलेशन इन प्रोटोप्लास्ट. तो प्रोटोप्लास्ट इज permeable for foreign genes like NEEF, disease resistant genes, hiding genes that we can easily manipulate or enter these genes on this protoplast, and then we can inoculate on this mutant medium. Then third is the induction of mutation. So in that induction, by using the physical or chemical mutagen, and that will be do the treatment of these mutagens on protoplast. So some mutations will be happening in case of the protoplast. Then addition of cell organelles in protoplast. So we can add some cell organelles like mitochondria or chloroplast carry additional genes for resistance for the antibiotic activity or fermentation, and that we can easily add these cell organelles on this protoplast, so they can create some variations in the plants. Then in next stage is the protoplast cultivation. So this is the treatment हो गई प्रोटोप्लास्ट पे. Then we can cultivate this protoplast. So for cultivations of the protoplast, the MS medium, mostly MS, that means Murashige and scoop medium, are used for development of the callus. These on this protoplast continuous division will be takes place and the callus are formed from the protoplast. Then this callus is callus means क्या होता है, एक mass undifferentiated mass develop होता है. Then the mask will be this mask will be re-inoculated or recultured on the separate test tube. In the test tubes contain 1 μm benzyl aminopurine for more shoot formation. कैलस का हम उसका रिकल्चर करेंगे. उसमें जो शूट फॉर्मेशन के लिए जो हेल्पफुल माइक्रोन्यूट्रिएंट्स है वो हम उसमें ऐड करेंगे. देन शूट का अच्छे से फॉर्म होने के बाद हम अगेन दैट विल बी रिकल्चर्ड ऑन द फॉर रूट इनिशिएशन. जो रूट इनिशिएशंस के लिए जो भी माइक्रो एलिमेंट्स ज़रूरी है वो उसमें उस मीडियम में हम ऐड करके फिर उसका रिकल्चर करेंगे. देन आफ्टर डेवलपमेंट ऑफ शूट्स एंड रूट, these plantlets will be transferred on the pot and they will be slowly hardening for the greenhouse techniques. So this is all regarding the protoplast culture. The applications that I already discussed with these previous videos. So this is all regarding the protoplast cultures. So now I stop here. In next videos we talk on the somatic hybridizations or how the or which are the techniques or importance of the somatic embryogenesis. So now I stop here.